So hi everyone, welcome to the December session of the Chai seminar. Today our speaker will be Tom Nuno Wolf. Nuno is a PhD candidate at TU Munich, and he works at the in primarily in explainable deep learning models for in medical image analysis, but also I think more recently for including image and tabular data as well. He's part of the VR Viz group are you part of you are part of the VR Viz group, I
You
believe.
used to be
Ah,
Lab for
sorry.
Artificial Intelligence and Medical
Um,
Imaging at
yeah,
the moment.
sorry. Yeah. But sorry about that. Yeah. But I won't take too much of your time. Thanks for doing this, Nino. And whenever you're ready.
Yeah, perfect. Thank you so much for the kind introduction. Okay, let me share my screen and then we'll start. All right. I hope you don't see this part yet. All right. Okay, so thank you very much for inviting me, first of all. So I am Nuno. I'm affiliated with the Lab for Artificial Intelligence and Medical Imaging, and we're affiliated with the um, Department of Radiology at the University Hospital of Technical University of Munich. I'm also affiliated with the Munich Center for Machine Learning. Um, and today I'm going to talk a little bit about interpretable neural networks for the differential diagnosis of Alzheimer's disease. And just a little bit of a scope, what we'll discuss today. First of all, explainable AI and like which parts of it are important for my work. Then I'll talk a little bit about a study we did on the faithfulness of case-based reasoning. This is a completely overused term at this point. We proposed some type of a definition, but it seems like every paper is proposing a new definition for this term. So yeah. And then I'll introduce a new method that we recently published for case-based reasoning and conclude with an application regarding the prediction of dementia with these types of networks. All right, so first of all, explainability. I think most of you are familiar with these techniques that can do pixel level attributions. This is what I'm concerned with primarily. And as you see here, we have pretty common methods, for example, GradCam, occlusion sensitivity analysis, or integrated gradients. And depending on the method that you choose, you will get some different highlightings of different features. You can evaluate these attribution methods and specifically the explanations they provide given these axioms, which are a sensitivity. So you want you want explanations to be sensitive, meaning non-zero. If the network depends on them for a forward pass, you want them to be implementation invariant. So if you have two different networks and their predictions are identical for all possible inputs, then you also want the explanations to be identical. You want them to be complete, which means, in a nutshell, you want all the explanations to add up to the prediction of your model. There's the dummy attribute, which means if your prediction is invariant of a specific feature, you want it to have zero attribution. Linearity, which means if your network computes a linear combination of two subnetworks, this linear combination should also hold for the attributions that are computed. And finally, symmetry preserving. So if two features have identical contribution and then you want to be able to replace them, and the attribution should also remain the same. All right, so big question is, how do we get explanations in general? And typically you have a black box neural network, you have your data, and you try to do some type of prediction. This usually involves fitting or using a surrogate model that creates the explanation for you. And Cynthia Rudin did this rather controversial statement in which she said post hoc explanations must be wrong. What she means by that is that if you have a surrogate model that perfectly explains your black box, why do you need the black box in the first place? Couldn't we just use the surrogate in a way? And this is motivation to inherently interpretable models, which not only predict do some type of prediction, but also provide explanations to you, typically at intermediate stages with the attempt of making them human understandable. A very common technique is case-based reasoning. This works as follows. Given an input image, you compare it to something called prototypes. these should be class representative patches. For example, here we see that this prototype should be similar to this part of the test image, and for the other ones analogously. And then we can reason as this part of this bird looks like that part from my training samples, because these prototypes are exactly training samples. Now, in addition to highlighting which area is similar, you can also compute attribution maps. And from these attribution maps, you usually derive the region of the input image that is important. Now, a very famous and popular implementation is prototypical neural networks, proto-PNet primarily, and they work as follows. So first of all, you have an encoder U. This creates a latent space embedding, specifically latent feature map Z in this case, with some spatial dimensions. And in addition, you have a prototype module. These prototype modules are a bit complex. I'll try to break it down easily. So you have a set of prototypes. These are trainable parameters. They're just vectors. Just assume this for now. And then if you want to do a prediction, you take each of these prototypes and compute a similarity between each of these vectors and the spatial vectors in your latent feature map, which yields a similarity map. Now, in order to classify the image, each prototype should have some different meaning. You do max pooling on the similarity map, which gives you an idea about the maximum response in the image for each prototype, which yields a similarity vector or a vector of similarities, which you can then feed into a linear classifier. Now, if you want to explain which parts of the input image were important, the original work proposes a pretty complex algorithm. In a nutshell, in modern neural networks, you upsample these latent similarity maps into the input domain, and then you overlay them with the input image, and deciding on some threshold, then you can decide which crop of the input image is the similar one to the prototype. You might wonder, how can we actually trace these parameters that are optimized during training to specific input samples? And what you do is you feed your whole training data set through the network, and you save the similarity scores for each of these patches to all of the prototypes, and then you replace the prototype with the latent feature vector from the training sample that it is most similar to. And again, you compute the explanations, crop through the region that's most similar to it, and then you know which parts of the input image is probably comprised in this prototype. All right. Now, this is it with background for now. So let's first look into this case study that we did called Proto-P-Faith. We wanted to use these architectures for medical applications. And what was a bit concerning at one point was that we believed, okay, can we actually guarantee that these attribution maps in latent space are traceable into the input domain? And in the general case of large neural networks, your receptive field in latent space is pretty much the whole input image. And this assumes that there is a spatial dependency between the input domain and your latent feature space. And we came up with a quick proof via counterexample that I'll talk you through. So we have an input here with the blue rectangle around it, and we convolve it with the weight vector in the first layer with stride 1 and padding 1. This yields this latent, and my mouse is gone, a latent feature map on the right. And then we feed this latent feature map into the second layer of the network with stride 2 and padding 1, and these network weights. And this gives you the bottom right feature activation and latent feature map. Now, if you upsample according to the algorithm, you would assume, or you would believe, that the bottom right of the input image is driving the decision. But because we know the network weights and all the parameters in the input, we know that the top left feature of the input actually drove the activation of the bottom right feature in latent space. So this is concerning in medical applications because you usually want to be pretty certain about the region that you are attributing to. For example, radiotherapy, you want to expose the not healthy regions to radio signals, but the malignant ones. We also evaluated the model under a lot of other axioms. Specifically, the linear classification head holds for all of them, but this upsampling here is problematic. Also, max pooling is problematic. Excuse me, a lot of these, some of the axioms are broken, for example, completeness. If you want to read into it, I recommend reading to the paper. There we do the in-depth analysis. But this is theoretical right now. And there's also this argument that neural networks might be localized in real-world training. So we were interested to somehow verify this experimentally. And the idea was to somehow extract different features from the same network. And for this, we borrow from Shapley value approximation methods, specifically from DASP. And this algorithm works relatively well with relatively little approximation error for high-dimensional inputs, even works for for images. And what we do is we compute Shapley value-based explanations on the similarity level, and we want to compare them to the original explanations as provided by Proto-P-net. So we're able to do this well, there was a lot of technicality and maths involved to make this possible. But ultimately, yeah, we managed to do it, and we now got these two types of explanations. At the top, you see the vanilla Proto-P-net explanations, which appear very localized. You see in the second and in the fourth column, the prototypes, and in the first or third one, the test image. And you might believe, okay, like here, the model looks at something like the ear of the dog, and here it's maybe concerned with texture. But then when you look at the alternative explanations, which theoretically are more reasonable, you see that a lot of the input actually activated these neurons and similarity scores. So this is problematic. We also evaluate this quantitatively using the deletion score. And in this study here, you see that the new alternative explanations seem to be much more sensitive to to deleting most important pixels, which means they are more reasonable in a sense. Okay, so what are the important key insights from this study? Well, these theoretical violations, they manifest in our experiments. These findings also generalize to other implementations of case-based reasoning and also things like GradCam, if you want to explain intermediate latent feature maps, so be aware of that. And lastly, of course, these explanations that I showed, they're very difficult to interpret because at least I have a pretty hard time making sense of this. And this is obviously something to address in the future, and exactly motivation to our next study that we look into, because case-based reasoning has shown in user studies to be very human understandable. So we wanted to build upon this idea, but have something that is more theoretically grounded. All right, so in this study, we looked at explanations from three different angles. They should be local, which means you want to predict an individual prediction of the network. They should be human intuitive. What we mean by that is like, don't show them a dozen of explanations, but as few as possible, and also global in the sense that you have an overall understanding of the model, and faithful, again, that in our case, this means satisfying these axioms. All right, and this is what we proposed. It's a large figure. I want you to concentrate on the center of it for now. So given a test sample, we map it into latent space. And if we want to do the prediction, we have, again, something called support vectors. Conceptually, they are similar to prototypes. The optimization is a little different. It is a little simpler to train. And then you compute, again, something like similarity scores between your supports for each class. You take the sum of these evidences to yield your logit of the class. Additionally, we can create explanations both on the support vector level, as here, and on for the test sample. If we create the explanations for different classes, for example, here for the predicted class compared to the Brittany class, you see that the focus of the model shifts quite a lot. So this indicates that the explanation is also very contrastive, which is what we want from them. If we look into other explanations, for example, from the support vectors, we see that the model only focuses on the dog and not on, for example, this, I think it's a wine barrel, or here on the right on this like toy on the dog's head. Okay, now 2 slides of maths for the background and the technical background of these methods. We build up on 2 prior works, one of which are the because transformations. The because transformations actually allow you to create these very pretty explanations. And in our work, we also prove that they fulfill these axioms. So this is very nice. Because transformations modify standard linear transformations as computed by any layer of typical neural networks. They extend them by introducing, first of all, an exponent b into your cosine similarity, which increases alignment pressure. The idea of this is that you have your feature vector or your features that you try to find in your input x, that weights that are poorly aligned just are more angle dependent rather than magnitude dependent. And you also normalize your weights. This is important to create the explanations later on. All right, and additionally, you remove all biases and nonlinearities, and this allows you to reformulate the forward pass of your neural network as a concatenation of linear transformations, as seen here. And now, concatenation of linear transformations can be summarized in a single weight matrix, and this is the very important property about these because neural networks, because each forward pass is now input dependent on your x, but you can compute this weight matrix, and then if you simply, in the end of your network, compute a linear transformation, you can create explanations that are exactly traceable to each input feature. This is the great thing. In their work, they also showed that these models can learn nonlinear dependencies, although they discard all nonlinearities like redo activations. The second thing we build upon is the Nadaraya-Watson hat. The Nadaraya-Watson hat is a non-parametric classification hat. You embed your inputs into latent space and then take, for example, your whole training set and the latent embeddings between them, and you compute the softmax to all of across the whole support vectors that you choose. Might be the training set, might be clustered with k-means. And then you essentially predict the class with highest evidence. All right. Yeah, so during training, you sample randomly from each batch, for example, because you need to optimize in a way. So what happens is you align different images from the same class to same direction in latent space. And this is our architecture and implementation. So like any case-based reasoning method, we encode support samples and our input image. This yields latent feature vectors in our case because we use global average pooling. We also encourage the model explicitly by making these vectors positive to be dependent on the activation of your class, but be positive. So you don't want negative correlations of some features be used to predict a specific class. For example, the presence of a tire should not decrease the probability of a dog. All right, and then we compute the similarities between the support vectors and our test sample, which yields again the similarity vector, and we take the sum across these similarities. And as seen in the previous slide, we know how to compute the explanations now. Again, here, a very contrastive example. If you predict the cat, it uses the cat. If you predict the dog, it uses the dog on the side of it. Okay, now let's look into more results because we have more dimensions than just showing these images. But the images are important in a way because we have the support samples and we can compute the similarity scores as seen before. In addition, we have support evidences, which are the similarity scores between each support vector and the test sample in latent space. I want you to pay attention because here you see that these scores are pretty similar for all of these three vectors. On the next few examples, we see something different. This is essentially an indication that the network learned a clear decision boundary for this class, and therefore, when we run the algorithm to compute the support samples, which is k-medoids, by the way, it essentially finds it doesn't really matter which support samples you choose because in latent space they're also similar to each other. So you can assume this as a bank of just exemplars that help you as a human understand which parts of some images your network uses. All right, and then, yeah, just like in all the previous ones, we see predicting the bottle uses the bottle and the dog. All right, we also evaluated the model on a medical data set, this RSNA data set, and we created a data set where the model has to predict opacity in the lungs. Without doing bounding box regression, it simply has to do a classification. And we use the bounding boxes to verify if the model uses features that are reasonable in a medical sense. Now, we see, for example, for this test image that the model indeed uses primarily features that are seemingly opaque. And looking at the support evidences, we see also that they are pretty different, which shows there's high variability of this class in latent space. Now, looking at the support samples here, we see for support sample number 4 and number 5 that they are also somewhat reasonable, I suppose. But for support sample number 3, we see that there's activation across the whole input image. And this is a little bit suspicious. Also looking at the support sample 0 of the healthy control class, we see something similar. So we were interested to see what this model actually might have learned in latent space. So we ran a T-SNE embedding, and in this specific mapping, we found that there's a continuous region of the two classes, which is an indication that the model did not learn a clear class decision boundary, and the prototype support samples that we identified are also pretty close in latent space. So what you can do now as a user of this method is you can decide what you do. Do you want to rerun your model? Do you want to adapt your normalization? You might also want to just discard these support samples that are not really, well, they are not really useful anyway, and you base your prediction on the, well, let's say more healthy support samples. Okay, but since we have these support samples, we can also compute interclass similarities and intraclass similarities, which is what we did on Pascal VOC. So you want interclass similarity to be relatively high and intraclass similarity to be high and interclass similarity to be low. But this gives you an idea about correlations and perhaps spurious correlations in your data set. It's well known that the Pascal VOC data set has some, well, the annotations are not so great for the labels. And using this similarity map, you can directly see which classes are correlated. For example, bottle and person. I can tell you from evaluating some images there are plenty of people having a lot of fun. So this is kind of expected. And then people sitting on chairs, things like that are quite natural, I guess, as well. We also see that there's a little higher interclass variability for the horse class, which means this class seems not so well connected. And in fact, we can look into this and see that there's an outlier in terms of supports samples for the horse class down here because the rest of it is clustered around here. We also evaluated the model quantitatively on 3 different data sets, or actually 4. It performs pretty much on par and similar to all the other explainable methods, interpretable methods, and also on the black box, depending on the task. Importantly, we ran it on the Funny Birds benchmark. This benchmark allows you is a collection of synthetic images and renderings of birds, and you can artificially just remove parts of this image. For example, you can remove the beak if it might be discriminative of the class for a specific bird, or you could flip colors of some feathers, for example, which might also flip your class label. And then having a collection of these flipped images, you can compute a whole variety of scores. For example, the single deletion score, which is the problematic score for our method, in fact, but actually all methods have a problem with it. And if you look at Protopinat, it performs pretty poor. Yes, and what are the key insights from this study? Well, we were able to create a case-based reasoning approach that is theoretically grounded. We've seen how we can use these global explanations for probing for alignment. We've seen in the medical study that you can directly see if your model learns something that's not as expected. And interestingly, these models are now also versatile across backbones and tasks. Well, we show this in our method, but they are pretty versatile now because the group that proposed because models recently published a paper that's called Becauseification, and they showed that you can convert any neural network into a because neural network and then fine-tune this because neural network version of it in just a single epoch and have pretty competitive performance. We also have the code available, so I highly recommend you to try this. Okay, so for the last few minutes, I would like to take you on a journey to the medical world and specifically the world of neurodegeneration. We want to do dementia prediction. I'm going to exemplarily talk about Alzheimer's disease, which is the most frequent dementia disease from all the syndromes that describe dementia. And it's expected that it will affect 150 million people in 2050. The disease progression is relatively unknown, and that's why huge flagship studies like ADNI collect a lot of heterogeneous data. For example, PET imaging, which will result in a three-dimensional volume, genetic testing in the form of identifying the mutations on the ApoE allele, and also measurements from the cerebrospinal fluid or blood measurements and whatnot. Demographic information plays a role. So they try to collect all of this data and then be able to infer how the model progresses over time. For those of you who are unfamiliar with PET images, here's an example. PET imaging works as follows. You give the patient a radio tracer. For example, here it's FDG, which is mimicking glucose. So it's, you give it to the patient and then you wait some time and then you measure over some time and you measure the absorption of this glucose mimicking molecule in the brain, just like with CT. And this allows you to create these 3D maps. Now, what we see here is that we have on the left a healthy patient, and we see that there's a lot of activity as you would expect. But on the right, there's a patient suffering from Alzheimer's disease, and here you see that in the temporal lobe, there's a lot less activation as compared to this healthy patient. So this is the type of data that we use in this study. Moreover, to give you more background, you see that the disease stages, which are assessed via psychological testing in clinical practice, here on the x-axis, cognitively normal, the stage of mild cognitive impairment, which is when you have first symptoms like you keep forgetting things, you can't memorize some words, and these kind of issues, and then ultimately dementia. And while structural MRI is pretty cheap and easy to assess, it usually changes relatively late in the disease progression. In contrast, these functional imaging methods like PET, for example, they change much earlier. All right, so what we want is an inherently interpretable neural network for this type of data, and we propose the prototypical additive neural network for interpretable classification of Alzheimer's disease, short PANIC. And we use generalized additive models in this study. So these methods are univariate functions. So you have a set of univariate functions. Each function gets one input feature from your tabular features and does some type of a prediction. They were extended in a study to neural additive models, which actually train neural networks instead of just linear functions. And the important thing here is that these neural additive models can be trained with backpropagation algorithm. So the question, of course, is how do we fuse the imaging part of our large heterogeneous set of data? And we leverage ProtoPNet. And now this summarizes the whole talk, why I actually looked into prototypical neural networks so much. Because in this initial study, we used an extension of ProtoPNet called the XProtoNet. We don't have to go into the details right now because these methods, they are pretty similar to prototypical neural networks. They just have a different way of computing the explanations, but the problems that we found for ProtoPNet unfortunately also transfer to these type of networks. Well, what we did was we extracted the similarity scores of these prototypes and treated them as univariate functions to append to our global generalized additive model. All right, and this method is pretty hard to train. In fact, in general, prototypical networks have pretty multitask losses. In this case, you see 5 different terms. They are not great to train, let's just put it that way. But they are glass box models. And I'm going to show to you now how we create local and global explanations with these methods. And I'm pretty sure you will like what you see. Because first of all, we have local interpretations. So here we see the effect of each input feature to the prediction of this sample being diagnosed as Alzheimer's disease. For example, the left hippocampus volume, which from experience I can tell you that this is a relatively low number, was driving the decision primarily, and then our image-derived similarity scores were also very important. And then here we have a set of genetic information, which I don't remember what they are exactly. But yeah, beta amyloid is also known to be a biomarker of AD, so that's reassuring. And then second, we can look at the attribution maps in 3D space. So first of all, I want to explain the prototypes. So we see which parts of the input image are actually comprised in the prototypes. Verifying this with clinicians suggested that there is some overlap with known regions of the disease. And then we also looked at the predictions, or look at the predictions for this test sample, and you see that there's some overlap between the prototype and the region believed to be similar to the prototype of the test sample. We can also compute global interpretations by computing the mean importance of each feature, and we see that the similarity scores are derived from the image, from the PET images, are most important usually. Left entorhinal volume, left hippocampus volume are known biomarkers of AD, so that's also reassuring. Just like beta amyloid and tau, they are well known. Also, age is known to be possibly confounder, but to play a large role in Alzheimer's disease. All right, and now to my final explanation map of this method, which are the log-odds ratios. So conceptually, what you do is because you have this set of univariate functions, you fix all other variables with the exception of the variable of interest, and you compare to some baseline, which in our case is the healthy control class. You modify this this feature, and this allows you to plot how the probability of being diagnosed with a different class changes. For example, we see here with the left hippocampus volume that with increasing hippocampus volume, you have a lower probability of being diagnosed with Alzheimer's disease or mild cognitive impairment than being healthy control, and with lower volume, vice versa. For the left entorhinal cortex, however, we see that towards the tail there's this weird increase in probability, and this is unexpected. Is problematic because usually you would expect people with higher entorhinal cortex to have lower probability of being diagnosed. But this is exactly this glass box property that we want from these models to really evaluate if they are clinically aligned. In this case, I believe it might be due to outliers in the data set. So what you can do now, you have this insight, what your model learned and which data distribution it learned. You can just look into your data set, look into the training samples that are in this range, and perhaps there is something that you actually might learn from the data. In general, in many cases of these top performing features, they are clinically aligned. But as you see also here for beta amyloid, there's also something where it's like a little unexpected, I would say. All right, so what are the key insights? We were able to create this interpretable model for image and tabular data, and we've also showed that they are pretty easy to evaluate for clinical alignment. This marks a very important step because, as I said earlier, clinicians are much more likely to use and trust these models if they can evaluate their clinical alignment. All right, what can we take away from this talk? So first of all, we introduced PANIC, which is this transparent classifier for image and tabular data. Then we proposed the users at the theoretic evaluation of prototypical neural networks called Keep the Faith. This rigor evaluation showed that there are some problems with it that can be fixed, and others that might persist. And finally, we proposed SIG, the similarity-based interpretable image classifier for theoretically grounded image classification, which we're looking forward to adapt to Panic and then have a tool that we can use in clinical practice, and hopefully other people can benefit from this as well. With this, I would like to thank you on behalf of all my collaborators on these projects, and now I'm open for questions.
Thanks so much, Nuno. Please do let us know if you have any questions. I think the question, I think there's the one which you might have already answered, I believe, the proto-PNet relating to information bottleneck.
Was it in the chat?
I believe so, yeah. So the question is, how does proto-PNet relate to the information bottleneck approach?
Okay, information bottleneck. Are you referring to concept bottleneck models?
Yeah, I believe so. That might be, yep.
All right. Okay, so assuming that, yes, okay, how do we compare concept bottleneck models and prototypical neural networks? Good question. They are, well, I think they have pretty different fields of application. So I would say you usually, first of all, you need annotations for concept bottleneck models. And then you can ground your prediction on essentially first having the model predict a set of concepts that you know are correlated to something. So for those of you that are unfamiliar with them, in medicine, you might want to predict some disease, and you have a set of features that you can extract from an image, say, I don't know, some fracture in a bone, that is very specific for this type of, well, disease. And then you have just like a linear classification again in the end of these predicted concepts. Personally, I haven't worked with them too much. I think conceptually they're interesting, but given the findings about attribution methods, I'm not sure if I think you have the problem that they only give you this supposedly explanation by saying a feature is present or it's not present that you know, but you don't know what the model used to predict this feature. For example, you cannot detect if the model learned spurious correlations. The method might predict some feature that you defined just because there are some spurious correlations in your data. And that's something that you will never learn with concept bottleneck models. But of course, combining them with because networks might be an interesting project for someone out there.
Right.
Oh, there are more questions, I believe.
Any other questions?
Oh, yeah, okay.
Well, in that case, maybe I'll ask a couple of questions here and there.
Yeah.
Tom, so given the SIG method that you kind of approached, and I think we might have briefly spoken about this, but just for the general public, what generally happens when you might have two classes which are very very quite like very very similar generally, and but obviously the classification kind of differs between the two classes. And what sort of do you kind of get a collapse of the explanation between those two given classes? Like for example, like you just have a healthy brain and like a diseased brain, but like the tumor is like really small. But then that is discriminative for telling like the brain is diseased or healthy. But like again, if you're looking at it visually, it might not be very like very like super perceptive. So do you kind of face like issues in those cases when you're trying to attain explanations?
Okay. So I think the whole method really depends on how well the separation latent space is between the classes. So if your neural network is able to somehow learn this mapping where you have kind of different clusters in your latent space for the two classes, then you can expect to have really contrastive explanations.
Mhm.
Um, the so I think it really depends on that. So if I think it's not necessarily a problem of the method, it's more a problem in general than with your data.
Mhm.
If you are able to learn such a mapping, then you should be fine. The way that the optimization works of this method is that since you collect some randomly sampled other support samples during the optimization, um, you strictly enforce to learn different clusters for each class. This is how it works. So at the end of the day, it's an optimization problem. Yeah.
Yeah, great. And the second, I guess, more of like a lead-up question to that is, given an example that you were trying to explain, and let's just say it was out of distribution for
Mhm.
the model that the sick network that you're trying to look at, is there some particular sort of signatures that you can see in the explanations or like in the
Mhm.
support samples, which will kind of give you an indication that maybe this is something that is out of distribution with respect to this model and therefore should not be like considered?
Yeah, that's a great question.
Yeah.
Unfortunately, we have not run these experiments, but I think that would be very interesting to see. So I can't just answer like hypothetically here. Um, I would expect that you can see, well, first of all, you can trace given the test sample, you can trace which input features drove the decision. So perhaps you see that some unreasonable features are being used for this prediction. Then you see in the evidence scores, you might see that there's a peak in that's unreasonable activation or just very low activation. So there's another dimension with which you could evaluate this. And of course, your support samples, they are fixed, so nothing would change for them.
Any other questions? Perfect. All right then. If that is it, then I would like to again formally thank our speaker. Thanks, Nuno, for taking time out to come and give the talk. I really appreciate it. It was very interesting, and just looking forward to see what you come up with next.
Yeah, thank you so much for having me.
Yeah,
Thank you.
perfect, perfect. Thank you, everyone.